Structural bioinformatics. The branch of bioinformatics that deal with the analysis and prediction of the three-dimensional structure of biological macromolecules such as proteins, RNA and DNA is known as structural bioinformatics. It deals with generalizations about macromolecular 3D structure such as comparisons of overall folds and local motifs, principles of molecular folding, evolution and binding interactions, and structure of function relationships, working both from experimentally solved structures and from computational models. The structural bioinformatics can be seen as a part of computational structural biology. Structural bioinformatics has existed in some form or the other ever since the determination of the first myoglobin structure. Structural bioinformatics has a significant role in the field of molecular biology. The main challenge for structural bioinformatics is the integration of structural information with other biological information to have a deep understanding of biological function. The success of genome sequencing projects has created information about all the structures that are present in individual organisms as well as both the shared and unique features of these organisms. Homology models for the structures identified through genome sequencing projects can be created with the help of structural bioinformatics. The resulting structures will be studied with respect to how they interact and perform their functions. Similarly, the emergence of microarray expression measurements provides an ability to consider how the expression of macromolecular structures is regulated at a structural level, including the reactions associated with transcription, translation and degradation. The alteration in functional characteristics such as structure variation, genetic mutation and post-translational modifications can be studied through structure modeling. Moreover, 3D structures of the biological molecules can be analyzed through this application. When the organization and physical structure of entire cells is understood and represented in computational models that provide insight into how thousands of structures within a cell work together to create the functions associated with life. In any case, in the field of biology, structural bioinformatics is very important because structure alignments provide us with more information that is not available from current sequence alignment methods. Structural bioinformatics is now in a renaissance with the success of the genome sequencing projects, the emergence of high throughput methods for expression analysis, and identification of compounds via mass spectrometry. There are now organized efforts in structural genomics to collect and analyze macromolecular structures in a high throughput manner. These efforts include challenges in the selection of molecules to study, the robotic preparation and manipulation of samples to find crystallization conditions, the analysis of X-ray diffraction data and the annotation of these structures as they are stored in databases. In addition, there have been advancements in the capabilities of NMR structure determination, which previously could only study proteins in a limited range of sizes. The solution of the malate synthase G complex from E. coli with 731 residues has pushed the frontier for NMR spectroscopy and suggests that NMR is having its own renaissance. Finally, the emergence of this structural information when linked to the increasing amount of genomic information and expression data provides opportunities for linking structural information to other data sources to understand how cellular pathways and processes work at a molecular level. Protein structure prediction, protein-protein interaction prediction, molecular interaction and protein-protein docking are some of the applications of structural bioinformatics. All these can lead us to have better understanding in molecular biology. Two commonly used databases for the structural data are PDB and EMDB. PDB or Protein Data Bank. The worldwide website of the Protein Data Bank at the Research Collaboratory for Structural Bioinformatics (RCSB) offers a number of services for submitting and retrieving three-dimensional structure data. The home page of the RCSB site provides links to services for depositing three-dimensional structures, information on how to obtain the status of structures undergoing processing for submission, ways to download the PDB database and links to other relevant sites and software. EMDB or the Electron Microscopy Data Bank. In 2002, the EMDB was founded at the European Bioinformatics Institute specifically to archive the non-atomistic structures determined by different methods like single particle methodology, electron tomography and electron crystallography. Today, the EMDB contains over 1300 released entries and is expected to grow 5 to 10-fold by 2020. Since 2007, the EMDB has been managed jointly by three partners: Protein Data Bank in Europe, RCSB PDB and National Center for Macromolecular Imaging or NCMI at Baylor College of Medicine.